Okay, the last part of these notes, I am going to go over all the stages of cellular or aerobic respiration for you. I am going to show you what I want you to know about it. There's more to know, much more, but that's for a majors level cell biology class. So notice I have a legend. In black, anything in black will be a substrate or a reactant, something that goes into that stage. In blue will be the end products of that stage. With a green box around it, that will mean that that compound is ETS bound. With a red circle, that's substrate level phosphorylation. With a purple circle, that's oxidative phosphorylation. These are two ways of adding that third phosphate group to ADP, adenosine diphosphate. So two ways of adding a third phosphate to adenosine diphosphate. Okay, phosphorylation is simply adding a protein. Now, ADP is not the only thing only compound that has phosphorus phosphate groups added. There are proteins that have that too, but we're not talking about that in this course. That's more for an anatomy and physiology course. And then the brown is waste. Now I am going to oversimplify glycolysis. So what I'm going to do is I'm going to draw 6 black spheres or circles, okay? These represent C6H12O6, or glucose, a substrate of glycolysis. Then, and this may not even be the correct number of arrows, but I'm showing multiple arrows. Do you see that? And those represent, each time there's a break, that represents an intermediate. We don't just go from glucose to pyruvate. It's not that simple. In fact, I urge you to look in your book. I believe it is probably in the appendix. If it's not, it's probably in the chapter. Oh, you need to read this chapter on cellular respiration. But you need to look in your book or look online and look at the full-on process of glycolysis to truly appreciate what's going on. But this is the simplified version I want you to know. So we have glucose plus, and the direction of arrows is very, very important. Notice these are going in. So it takes some energy to get a little bit more energy in glycolysis. So the energy is in the form of ATP. There are 2 more compounds, NAD. In your notes, I've shown you what NAD looks like chemically. And I'm not even using the name that NAD is standing for. I think of these as hydrogen and electron taxis. Okay? Hydrogen and electron taxis. NAD. Okay? So there are 3 substrates. Glucose plus 2 ATP plus 2 NADs with pluses. And that would be like a taxi that had some vacant sign. That's what the plus is like. It has been oxidized. Yes, it has lost an electron or electrons. Okay, then at the end, if these circles represent our carbons, they're all still there. Okay? We had 6 here, we have 6 here, they're just in 2 packages. So basically we have 2 3 carbon pyruvates. I've put the chemical structure of pyruvate in your notes. You don't have to know that, but you can look at it if you'd like to see that it truly does have 3 carbons. The NAD pluses are reduced. In other words, the passengers get in to the taxi. Okay? The electrons and the hydrogen ions. And then, try not to erase things as I go. Notice the arrow is going toward making ATP, not away from it, so we didn't use the ATP. Notice there are 4 ATPs here. Okay, I need to go back to my legend, and I need to put my green square here, because we're not done with these NADHs. They have chemical potential energy in them. It's because of those added electrons and hydrogens. So we're not done with them, okay? But we have perfectly good ATPs that are not structurally different from the oxidative phosphorylation ATPs. They're the same ATP, they're just made differently by substrate level phosphorylation. So basically part of this is adding phosphates to intermediates, and then those are taken off. Energy from breaking down the glucose this little bit so far is used to make some ATP. Okay? Not created, not destroyed, just transferred from the glucose to the ATP. Okay, in that video for your discussion, I forget his name, but he uses Krebs cycle instead of citric acid cycle, and he puts the preparation for the citric acid cycle in with the citric acid cycle and really reduces it down to what bothers me. I want it to be pronounced and to show up. So the prep for citric acid or Krebs cycle. This happens twice for each glucose. And I hope you understand why in just a second. What I'm going to do is I'm going to take a pyruvate. Hey, what was an end product here is now a substrate here. And I am going to have coenzyme A come in. So let me write over here, CoA equals coenzyme A. Coenzyme A is a fairly large organic compound. Okay, I just need you to know that. Then I'm going to show an arrow going out. No, I'll wait for that, sorry, because I want all this in black. Okay, notice my arrow. I start with an NAD. So I start with pyruvate, coenzyme A, and NAD. What do I get? NADH. This is acetyl CoA. Acetyl CoA. And CO2. So if you've ever wondered why you do this, it's to get rid of these CO2s. That's why you do that. Okay? So we have another NADH that is ETS bound, and we have CO2 as a waste. Notice that there's no ATP produced in this stage. So we say there's 2 turns of the Krebs cycle or citric acid cycle for each glucose. Just like this preparation happens twice. And you know why that is? That is because there are 2 pyruvates. So basically, in glycolysis, we lost a couple, remember C6H12O6, a couple of the hydrogens have been broken off, some energy harvested to make ATP, and the 6-carbon compound has been split into 2 3-carbon compounds. And then in the prep for the citric acid cycle, some people include that in the cycle itself, the 3-carbon pyruvate has coenzyme A added to it, another hydrogen taken off, and this time, look, one of the carbons. So by the time we've done this twice, we're down to 4 of the original carbons from the glucose. Okay? Now in the citric acid cycle, OA equals oxaloacetate. Okay, so we have OA, we have acetyl-CoA. This has multiple intermediates. We have 3 NADs, pluses, so they're empty, and a new one, FAD. We have ADP plus P sub I down here. Okay? So our substrates are oxaloacetate, acetyl-CoA, 3 NADs, 1 FAD, and ADP plus P sub I, or just ADP if you want. Now, what do we get? So this is a 4-carbon compound. And when these 2 carbons that were from the glucose via the acetyl-CoA join that, or bound to that, that forms citric acid. Okay, so that's the first intermediate, but you don't have to worry about that intermediate. But then with the second intermediate, one of the carbons from the from the acetyl-CoA is popped off. Then we have the other one. So by this time, all of the carbons that we started with after 2 turns of this are gone. They're gone. That cellular respiration is the complete breakdown of glucose to water, CO2, and then the energy goes into ATP, except that is lost is heat. We're going to get an ATP. The coenzyme A is going to be popped off. We're going to end with oxaloacetate, and we're going to fill some taxis. Okay? Going back to our legend, substrate level phosphorylation. Waste. ETS bound. And I put the blue line under oxaloacetate, and this is what makes it a cycle, is you start with oxaloacetate and you end with oxaloacetate. So that makes it a cycle. I'm going to put a couple of or I think they're in the notes, but even if the animations are in the notes, I'm going to add them separately in the module section so you can easily watch them, because me drawing this on the board does not do justice to what's going on. And we still have the ETS. Oh my goodness, the ETS. Scoot this over a little. Or ETC, electron transport system or electron transport chain. And this is what we, okay, so actually glycolysis happens in the cytoplasm of eukaryotic cells, and the rest of this happens in the mitochondrion. In prokaryotic cells, it happens along cellular membranes, infoldings of cellular membranes. So keep that in mind. So this basically along that membrane, we have these proteins. Okay? So ET